All right. Thanks so much, Amir and Peter, for the opportunity to be here. Really incredible talks this morning. Looking forward to the agenda this afternoon as well. For those of you I haven't met, my name is Dionne. I am a professor of material science and engineering at Stanford. I also have a courtesy appointment in radiology, and I'm also a Chan Zuckerberg BioHub investigator. Our lab is actually kind of just diagonally across the quad. So if you're around during the break and want to have a tour, I have some students here who are more than happy to show you our capabilities. And broadly, our lab focuses on the science of nanophotonics, how to control light at the nano and molecular scale, both to sense and sequence molecules, and then also to synthesize new molecules. And a lot of times students will ask me, did you always know you wanted to be a scientist when you were young? And the answer was no. When I was young, I grew up in Rhode Island, and I was actually inspired by special agents Fox Mulder and Dana Scully. Watching the X-Files, I thought I wanted to be a paranormal researcher. And this is back in the day before there was internet. So I just went to the bookstore and started reading up on the paranormal. And alphabetically, that is right next to PH. So physics, started reading about quantum physics and became fascinated and hooked. In my spare time growing up, I also used to go ice skating on the streets of Rhode Island. Sometimes my students will still entertain me by going disco roller skating. And then I eventually moved westward. I was excited to somehow wind up in Hollywood, but I got called by a nearby casting agency, Caltech. That's actually where I met Amir. He and I were grad students together. And along the way, got to meet some really incredible science stars like Stephen Hawking and Jamie Adam from the Mythbusters. And then started at Stanford over 15 years ago now, and it's been a blast. If you do go on a lab tour, one really cool facility to check out would be the environmental transmission electron microscope. This is actually a capability that's here in this building as a shared facility to both internal and external users of Stanford. And with sub-angstrom imaging resolution and also spectroscopic capabilities, we can look at how materials are interacting with their environment. So in our lab, especially related to biology and quantum biology, we like to look at nature and how nature synthesizes different chemicals, for example, ammonia. And then gaining insight from those sorts of enzymes like nitrogenases, try to create chemicals at more ambient conditions using excited state properties, like using light. So definitely come check out those tools. Today, I won't be focusing on that work. Instead, I'm going to focus on some of the work that Amir set up my presentation so perfectly for, which is how we can develop methods that allow us to get data that is either almost impossible to get with existing tools right now or extremely slow to get with existing tools right now. And if you think about biological complexity, in humans, we have about 20,000 protein coding genes, but that gives rise to millions to billions of possible proteoforms, including their like post-translational modifications. And yet most of the data we have in biology is at the genomic and transcriptomic level. So how can we get more information about the working molecules of biology? And that really is kind of the thesis that's driven the last handful of years of my lab's work. On scattering and use spontaneous spectroscopy. So we come in with an incident laser, and then we use the molecular vibrations as a way of essentially changing the color of that incident laser, and then the scatter light becomes for whatever biosample, whether it's a molecule or a cell that we are probing. And I think historically, the two challenges with single cell Raman spectroscopy, and that's where these photonic sensors comes in, and doing that in a manufacturable way, so not just relying on, say, metallic nanoparticles that have 1 nanometer gaps. The other challenge with Raman spectroscopy historically has been the interpretability, and that's where advances in AI and ML have really come a long way and allowed us to now kind of dig into the Raman spectra and understand what its components are. So first, on the sensitivity side of things, how is it that this particular silicon chip works? Amir showed you exactly this figure, so a perfect one to have set up the presentation on. The idea is we have an SOI substrate, or in this case, a transparent substrate like silicon on sapphire, and then we illuminate that substrate with usually near infrared light, anywhere from 785 nanometers up to about 1100 nanometers. And then we have a series of nano antennas patterned on the chip that are designed to essentially concentrate light, but then also scatter that light as a dipole to the far field. So we detect basically on an imaging CCD the far field scattered light from each of these nano antennas. And if you think about Raman spectroscopy, there are a few ways you can kind of break down what matters most, but essentially you have to think about the electric field at the pump wavelength, the excitation wavelength, and the intensity, and also the electric field at the probe wavelength, or the Stokes scattered wavelength. So Raman spectroscopy usually scales as e to the fourth, because you have an e squared at the pump wavelength and an e squared at the probe wavelength. And if you think just about the e squared, you basically have this Q over V term. So you want to nominally have a high quality factor, but you also want a small mode volume. That's what the V stands for. So these particular nanostructures that I'll share in this presentation kind of provide a design framework for how you can boost the quality factor while also keeping the mode volume small and sub-wavelength. And these are all silicon dielectric nanostructures that, in this case, have kind of three core regions that define the entire resonator. Region 1 is what we call the cavity region. That's where most of the light is being confined. And then we have photonic crystal mirrors on the edges that make the structure look as if it's infinitely long, even though it is a finite length structure. So how do these particular structures work? If we look within that cavity region, that L1 region, like I mentioned, there's silicon blocks. Each of those silicon blocks supports electromagnetic modes called me modes. And if all of the blocks were to have the same width, and you were to illuminate with free space light, then you can see the electric field of light would basically oscillate between plus and minus 1 in each of those bars. But if we slightly vary the width of each of those bars, we take that normally incident free space light and we actually couple it into an in-plane mode, a waveguide mode. So that waveguide mode is orthogonal to the free space light, which is why you don't couple to it without the perturbation. But by having a perturbation, you basically get light to interfere between the me mode and the waveguide mode. And then you can see the electric field get strongly amplified. And in the far field, the scattering intensity spectrum has a really sharp resonance. It's a phonotype line shape that comes from the interference between the incident light and the quasi-bounding continuum mode. The quality factor of this particular kind of quasi-bounding continuum mode, or phono-like line shape, is inversely proportional to the difference in width between neighboring bars, so delta D squared. Of course, the smaller you make that delta D, basically the more finely tuned your setup needs to be to come in exactly a normal incidence. But with this particular structure, assuming you were to have essentially infinite periodic or infinite periodicity, you can get quality factors, into the billions or hundreds of billions. As you start now to reduce the waveguide length, here you can see now we're coming in with free space light. We've got this waveguide type mode that is being excited. It's scattering like a dipole into the far field. As you reduce the waveguide length, of course, you're going to have scattering out of the end facets of this particular waveguide, and that scattering is going to kill your quality factor. So that's where we have those photonic crystal mirrors that come on the edges. And there's this really beautiful paper now from over 20 years ago by Noda and colleagues, where they have this quote, confine light gently to confine light strongly. Basically, you just want to have mode matching between the mode in your cavity region and whatever the modes are in your photonic crystal cavity, such that you can still maintain high quality factors. And then you can see in this particular example structure, most of the mode is confined within that cavity region. You can also play lots of tricks with slots. And here's just one example where the slot was kind of drilled through the center of the entire structure. Those slots, kind of a la Mikhail Lipson at Cornell, help further reduce mode volumes and basically give you kind of a silicon insulator or silicon air, silicon waveguide in the middle. And now you can get mode volumes that are below 1/10 of the wavelength of light cubed. So here's just one example showing the slots kind of through the center, where you can see you get a strong boost in the electric field intensity, but very small mode volumes while maintaining the high quality factors. And there are lots of tricks you can play with the slots to basically further reduce the mode volume. And then, like Amir mentioned, you can pattern many of these on chips. So in our first paper that came out about a year ago now, we had quality factors that were in the thousands, and we patterned about 5 million sensors per square centimeter in Stanford's clean room. We can individually address each of those sensors using bioprinting. So for those of you who have done biology before, the number of sensors we have and pretty much the number of assays we can do on chip would be equivalent to over 31,096 well plates. So this would usually take up the size of a basketball court, but we're really miniaturizing kind of the number of measurements we can do on a single chip. And then in addition to being able to reduce the mode volume with these slots, you can also get quality factors that exceed millions by, instead of having width perturbations, having height perturbations. And that's because in clean rooms, we can more precisely control the height, for example, using atomic layer deposition. So using height variations and also refractive index variations, my student Daryl has now made some really beautiful structures where it's tough to see the height variation, but perhaps you can see the white outline of every other bar because it's charging a little bit differently in the SEM. And this was a calculation, but experimentally, we now have quality factors that are over 500,000, approaching 1 million, just based on how steady our experimental setup is in the lab. We're actually measuring vibrations of the building. And then, like I mentioned, these structures also have very small mode volumes and importantly scatter like a dipole into the far field. So if you look at the sort of data we collect from these chips, here's an example where what we're doing is basically looking at the wavelength scans of different individual resonators. So even though there's no fluorescence here, each of the silicon resonators is lighting up in the far field, and we are getting a direct readout of what the local chemical environment is over each of these resonators. So in this first paper, what we did is basically locally pattern each of our resonators with different polymers, in this case, kind of spelling out the Stanford logo to be able to look at different refractive index changes. So what we do is we put down local surface chemistry that allows us to bind non-specifically to different molecules, proteins mostly in this presentation. But here we just worked with a simple sugar just to see if we could actually collect Raman spectra within these cavities at the single molecule level. And if you scan the polarization, also if you scan the pump wavelength, you can see basically the Raman signal of this sugar goes up where you would expect it to when you're on resonance with the pump. And then if you look at all of the experiments, which now we're collecting essentially simultaneously within our field of view, they agree quite well with theory. And for our theory, what we did is basically combine DFT and molecular dynamic simulations to understand the vibrations of the molecules we're looking at. So you can do first principles calculations of Raman spectra. And these particular sugar molecules here actually have two different conformations. And from calculations, it's predicted that this molecule spends about 39% of its time in one conformation and 60% of its time in the other conformation. And we found that our experiments actually match up to basically a weighted average of each of those two conformations. So although not included in this presentation, one thing that I think will be really exciting is starting to do actually dynamic measurements of molecules in these cavities and potentially understanding from the Raman spectra the structure. So I've been speaking for about half of my allotted time. Maybe just to wrap up the physics of how these sensors work, when I think about the landscape of previous optical sensors, they either have an extremely high quality factor, which allows you to boost the sensitivity. That's basically controlling how long light spends interacting with a particular molecule. And there are basically examples of really amazing high Q factor cavities, including, for example, ring resonators or photonic crystal cavities. But it's really hard to pattern many of those on chip and also have that dipole-like scattering into the far field. And then if you think about other structures that do have that spatial or diffractive control, plasmonic or MEV resonators are amazing. They scatter like a dipole to the far field, but usually they have quality factors of order 10 or less. So our work kind of combines the best of both worlds, where we have a high quality factor, but also a low mode volume. We can fit many sensors on a chip, and then this is amenable with large scale fabrication. So before I get into the biology, I just want to give a shout out to some of my students and postdocs who are here, and some recent alum. Sahil, who recently graduated and now is at Psi Quantum, used to have this quote, "What can high Q do for you?" But he did some really beautiful work integrating these sorts of silicon resonators with electrodes and also with lithium niobate to make gigahertz rate electro-optic modulators. And one of his lab mates, Dionne, is now here continuing this work. My postdoc, Feng, has done some really beautiful work integrating these high-Q cavities with two-dimensional materials to create novel photon sources and optically addressable spin qubits on chip. And then lastly, Remy, who I believe will be here in this afternoon, has started to use these sorts of metasurfaces for chirally selective synthesis. So in this presentation, I want to give you two examples of how we've started to use these sensors for biology. And the first of which is de novo molecular sequencing, focusing in particular on peptides, which are protein fragments. Certainly by no means at the single cell level. I think what Amir is doing at the single cell level is pretty amazing. That would be, I think, a long-term goal of this project, but we at least can look at hundreds of thousands of cells and start to interrogate the milieu of peptides that are on the cell surface. And then I'll shift over to applications to cells and how we can use this sort of method to, at the single cell level, start to understand different phenotypic signatures of bacterial and mammalian cells. So on the protein and peptide sequencing space, what inspired me is really kind of diving into immunology. To be candid, during the pandemic, I was sitting there probably a few days after we were at home and was thinking, gosh, I'm a scientist. I want to be able to help with the pandemic in some way. So I had some really great colleagues over in clinical microbiology and immunology and asked them for good example textbooks. So I just started reading immunology textbooks. And essentially all of our nucleated cells have protein fragments expressed on their surface that are the beacons for our immune system to know if a cell is healthy or diseased. So essentially, if I were to boil down simply how our immune system works, or how part of it works, your T cells are kind of scouring around looking at these protein fragments called peptides or antigens to figure out if a cell is healthy or diseased. And if it sees a mutated peptide, basically that T cell is going to basically punch a pore into the cell and then introduce different chemicals that target it for death. And this paper from Nature Cancer, highlights that each T cell receptor, TCR, can bind to a particular, albeit unknown set of antigens, thereby defining T cell specificity. So if you think about protein fragments or peptides, they're comprised of 20 amino acids. Most of these peptides have lengths anywhere from 9 to 25 residues long. So the combinatorial space is huge, and it's extremely challenging to figure out what those protein fragment sequences are. So the idea we had is basically to take our high-Q small mode volume resonators, get the peptides off of the cells, and all of the peptides, regardless of their sequence, have a C-terminus. So we can use established chemistry to bind those peptides via their C-terminus to our chip. And then what we do is come in with chemicals, usually they're called Edman reagents, that allow us to cleave the peptide one amino acid at a time from the N-terminus. And then we dynamically collect the Raman spectra. And what allows for this to occur is the fact that the Raman spectra of individual amino acids is readily differentiable. And so you can build like machine learning models that basically allow you to look at the N-length Raman spectra for a short peptide and compare that to the N-1 length spectra, the N-2 length spectra, and basically in a fully de novo way determine what that sequence is. So we have our chips, and then we basically built a robust flow cell around it that can handle the chemicals of this Edman degradation process. And here's an example, basically showing a cleavage of a short major histocompatibility class 1 peptide, where you can basically see upon each cleavage event, you have a change in the Raman spectra, and we've built machine learning models that allow us to uniquely identify basically what that cleaved peptide is. And you can see the first order, like even if you're just doing something simple, like looking at the differences, you can basically see how the change looks very much like what you would predict it to be based on even just the amino acid sequence. So we spun out a company based on this idea called Pumpkin Seed, and we have been collaborating with Genentech, where they are very interested in creating new vaccines against metastatic cancer. The idea being that the first time you have cancer, you are likely to survive, but it's usually the metastatic disease that patients succumb to. And in their use case, what they need to be able to detect are just single point mutations, so single amino acids that differ between different peptides that basically are the drivers of disease, right? So those protein mutations are driving disease. So here they gave us, where they knew the ground truth, basically 40 amino acid sequences, 20 of which were the wild type, so the healthy peptide, 20 of which were the mutated version. And then we didn't know what they were, so we had to put them on our chip and figure out if we could determine what their sequence was. And here what we did is collect Raman spectra. I'm projecting it onto a two-dimensional plane. And you can see each dot corresponds to an individual Raman spectra. And when you project that Raman spectra, that high-dimensional information-rich spectra onto, in this case, a UMAP, you can see each dot of its own peptide kind of clusters together. There's some spread, likely because of the variation in conformation of each peptide, or maybe a little bit how it landed on the particular sensor. But for the most part, the wild type mutated pairs cluster closer together than other wild type mutated pairs. And they're also readily differentiable. So you can build an ML model that allows you to determine basically what the full sequence is. So here I just showed you the Raman spectra of the full peptide, but then you can do full sequences. So we've done this now for both 8 to 12 amino acid long peptides, where we have over 96% accuracy. And we've also done it on MHC class 2 length peptides that go up to about 20 residues. And in this particular case, there are actually phosphorylations at different sites, which from my understanding is very challenging to do with mass spectrometry. So with our company, we've been working closely with IMEC, a foundry in Belgium, which has been able to take the basically chips that Jack did during his PhD that were just like 1 centimeter square, and then expand that to now 12-inch wafers. It's a multi-layer stack. So this is not quite the full stack, but getting closer, and you can see they also look quite beautiful. Okay, so in the last, I think I have maybe 4 or 5 minutes, how can we go from molecules to cells? And on the one hand, cells are easier because you have more molecules to detect. You're not trying to do single molecule sensing. On the other hand, they're more complicated because they're kind of like a water balloon of molecules, and you need to figure out like how can you get meaningful information from this very complex Raman spectra. And what helped to motivate this project was a very close collaboration with Amanda Karane, a melanoma surgeon over in the School of Medicine, where she's been pioneering immunotherapy responses and treatments with her patients, but has found that there are basically right now no predictive biomarkers of outcome. So there's targeted therapy and there's immunotherapy. With targeted therapy, usually people look at different genes, or maybe a few protein biomarkers, and they say, okay, you will respond well to this particular targeted treatment. But with immunotherapy, there's no good way of actually predicting if patients will respond well or have severe adverse events. And in about 30 to 50% of all patients who get immunotherapy, they actually have pretty severe outcomes. So again, we use our chips, and kind of the goal is basically to be able to map out the tumor immune microenvironment. So the cancer cells, the immune cells, whatever the milieu is of cells, and then see if we can provide meaningful predictive power to be able to tell Amanda and others like her how a patient will respond to immunotherapy. So we started this work initially looking at bacterial cells just because they are smaller and simpler. What we found is that when we did single cell Raman spectroscopy and collected tens of thousands of examples of each individual cell's Raman spectra, we could build machine learning models that allow us to basically predict what the bacterial species is. And here we looked at 31 bacterial species and strain. You can see that in the early days, this was now over 5 years ago, there were some misclassifications, but the misclassifications of different bacterial types were actually within the same colored grouping. And here the colored grouping corresponds to the first choice antibiotic that would be prescribed to a patient. So we're like, oh, that's kind of neat that we're getting misclassifications in the Raman spectra, but within the same drug susceptibility grouping. So this was on cell lines. Then we started looking at patient samples. And the nice thing is you can keep adding more data into your model to improve basically the predictive accuracy, including, for example, on patient samples. So then given that we had this insight that potentially we could predict antibiotic susceptibility, we started working with tuberculosis, which takes about 40 days to culture. And you need to culture it to have enough bacteria to be able to put down antibiotics and figure out if it's responding. So what we did is basically make antibiotic-resistant strains of tuberculosis, in this case, a cow TB, trained machine learning models to recognize the Raman signatures of different antibiotic-resistant MTB strains. You can see they cluster together quite nicely, so you can build nice models that, with over 98% accuracy, predict the resistance. And then the cool thing is that the Raman spectra is not just a black box. So computationally, you can remove different peaks. And if you remove a peak and the accuracy of your model now drops down, or the accuracy of your prediction drops down, you know that peak is important for classification accuracy. So in this particular case, the peak corresponded basically to mycolic acid content in the cells. And then we chatted with our friends who were actually TB experts, and it turns out the mycolic acid content is changing in tuberculosis. I'll skip over this slide, but then now tying this back to the tumor immune microenvironment, what we did is look at different mouse cell lines and different human cell lines, and kind of did the same sort of experiment that we did with tuberculosis. So look at different cancer cells and then figure out, of the cancer cells that are persistent after treatment, i.e. the resistant ones, can we differentiate those? So here, these are UMAPs, where again, each dot corresponds to a Raman spectra. So basically hundreds of thousands of single cell Raman spectra. And then we dove into the model to basically figure out what are the wave numbers that are most important for predicting basically drug resistance. And then in that tumor cell, you have not only, or tumor sample, you have not only cancer cells, but also immune cells. So then we need to know, can you use the Raman to look at different immune cell activation states? And you have your innate immune system, which includes, for example, macrophages. You also have your adaptive immune system. Here, I just showed you T cells. Basically, like, these immune cells are not created the same. They're all kind of different, and they all have different phenotypic signatures that correspond to whether they're activated or exhausted or pro-inflammatory or anti-inflammatory. So here's just a high-level analysis of probably 2 students, whole PhD projects, really understanding, can we look at not only differentiating immune cells, but looking at different activation states of immune cells. And then putting that together, this is my last science slide, we were able to show that basically we could combine Raman spectroscopy and get not only the same information as transcriptomics can, but actually more information that can predict immunotherapy outcomes. So what we did is train models on patient tumor samples that were from Amanda's bank, and in 30 of 33 patient samples that the model had not seen, we were able to accurately predict immunotherapy outcomes. So I will wrap up, and although there was not a ton of quantum in here, apart from, I guess, single molecule Raman, maybe that's within the realm of quantum, hopefully you are walking away with the ideas that I think nanophotonics can shed new light on biology. I think there's a really exciting future for using these high quality factor small mode volumes for biosensing. I know Amir and others have also done some really beautiful work on optomechanics, and I just kind of snuck in that we're seeing basically vibrations of our building when we're doing these measurements. So I think there's also a cool chance to maybe do some optomechanics with these. And what we've done is both develop basically these CMOS compatible high quality factor sensors that have millions of sensors per square centimeter, and then use those for peptide sequencing as well as single cell phenotyping. So with that, I would like to thank all of the students and postdocs who contributed to this work. And thanks to our funders, strong overlap with everyone Amir mentioned. So yeah, big shout out to NIH. I hope they continue to fund us. I have many zombie proposals, as my colleagues would say, not dead, not alive, just not funded yet. But also a big shout out to the Chan Zuckerberg BioHub, the Moore Foundation, and the Gates Foundation, and happy to take any questions.
Fantastic talk, really exciting, and thank you, very clear. I am actually wondering, I know you were short on time, wondering about your ML methods. Can you say more about the methods you use, more about the effective dimensionality of the data structures that you extract? I'm very curious in its relation to neuroscience, quantum, and sort of other applications, in particular of the Raman fingerprint on your mutated peptides. Yeah.
Yeah. No, great question. Handful of years ago, we were using basically convolutional neural nets. We've also used like random forest boosting machines. All of our spectra, we basically do like baseline removal, or subtraction. But we go from, I want to say, about a 1600 wave number spectra down to roughly about like 100 features that provide kind of high accuracy classification for most of the problems we've looked at so far.
Okay, so it's about an order of magnitude reduction in the underlying space. Okay, thank you very much.
Yeah, amazing work, Jen, as always. I was really impressed by the TB stuff and very surprised to hear that it's a 40-day incubation period currently for TB. Why is it so much longer than with other bacteria?
Yeah, that is a great question. So yeah, we've closely collaborated with the clinical microbiology lab on campus. So they don't see much TB, if any, here on campus. And usually it's about a day and a half to 2 days to do the conventional antibiotic susceptibility test after culturing. Tuberculosis is actually a much longer bacteria than normal. And because of that, basically you wind up kind of increasing the amount of time it actually takes for it to divide and replicate. So usually of order 40 days to get enough bacteria. I will say there are some like PCR tests that can give you an indication. So you can get information much faster, but usually it's just kind of single point mutations that are driving the drug resistance. So those tend not to be as accurate.
Yeah, I've actually had that test before and it was quick. So,
okay.
Yeah.
Yeah.
And negative.
Good. I'm glad. Yeah.
Great talk, Jen. Great talk, Jen. Promise I pushed the button. Okay. Great talk, Jen.
3 times. I love it.
So really super small, not plasmonic small, but still pretty small neurons that are still bigger than these short peptides. The mode volume still is not homogeneous, right? You should have a higher field density, like immediately above the device exposure later on. And because as you're doing the edit degradation, when you're going from kind of farther away to closer, you see those effects.
Yeah, I'd say, okay, so our mode volume, basically it's about a 10 by 10 nanometer region. And then the mode volume extends, it's an exponential decay. So roughly about like 50 nanometers into the far field. Our peptides are pretty short, like MHC class 1, they're only up to about 12 residues, class 2. All of that we can do kind of in a label-free way. And I'd say as you drill down, you get higher accuracy in part because of the mode volume, as you pointed out, so you can kind of see that exponential decay. And in part because you've just seen that many more times, right? But then what the company, Pumpkin Seed, is doing now to go to kind of longer proteins is basically during that Edmond degradation chemistry, there are certain molecules that you bring in to degrade and basically cleave off the N-terminus. And you can look at how the Raman spectra shifts outside of the peptide fingerprint for whatever the terminal amino acid is based on the binding of the Edmond reagent. And then the nice thing is those can have large Raman cross-section. So then you overcome the challenge of the exponential. Okay.
So I think in the interest of time, maybe one more question. And
thank you, Jim, for
have a very long lunch afterwards. So lots of time for discussion.
Okay. Thank you, Jim, for the beautiful talk. So I was fascinated to see the 9 orders of magnitude higher bit rate for biosphere relative to the technosphere. How many of those orders of magnitude is due to the channel, number of channels, and versus the SNR advantage? I wonder if the techno communication has something to learn from the biocommunication, if on a per channel basis, it has some advantages.
And yeah, I actually, I agreed on that calculation, but I want to give credit. That was not a calculation done by my lab, and they were looking mostly at protein. I think it's a really cool paper and one that invokes exactly the sort of questions where you're asking, where it's like not just about the number of channels, but
Right. Right. I'll get that paper from you. I hope that they could relate this to the channel, Shannon channel capacity theorem. Okay. All right. Thank you. Fantastic. Thank you.